Hi everyone, my name is Anushka and we're from the Amani Lab. Presenting our project, which is called the development of synthetic aptamers for use as low-cost PLD1 inhibitors. I'm Anushka.
My name's Kavya.
My name is Isna.
My name is Divya.
Okay.
Okay, today we will go over the purpose of our project in creating an aptamer, our methodology so far, the results we've obtained, what we've gathered from our results, and our acknowledgements. When starting cancer research, it's important to know the background of cancer itself. Cancer is one of the most dangerous and deadliest diseases in the world, as it affected millions of lives in the past and continues to affect millions of lives in the present. Could you move to the next slide? Oh, yeah. Though cancer comes in various forms and can be caused by multiple reasons, it is ultimately due to our body's uncontrolled cell proliferation, which allows for the development of abnormal cells, further destroying healthy tissue in our body. The two main types of treatment for cancer is chemotherapy and immunotherapy. Next. Chemotherapy consists of drugs which target the rapidly growing cells within our body, with its cost typically ranging from $10,000 to $200,000, which is relatively much less than immunotherapy. Immunotherapy, on the other hand, promotes the use of a patient's own immune system, and though that might seem like a better alternative, it is actually very experimental, which creates a risk in a patient's well-being in actually getting better while paying a minimum of 10 times more than chemotherapy or $100,000 in minimum, which can increase depending on a patient's specific needs and how their body reacts to the treatment. Because of this, patients are not always guaranteed coverage of health insurance. And this is why we believe it is important for patients to be able to choose between both options, especially chemotherapy. In our project, we focus on a specific protein, gene, and enzyme in our bodies, PLD1, which, once overexpressed, can create a roadblock for chemotherapy, which is why the goal of our project is to create a low-cost synthetic aptamer for use as a PLD1 inhibitor, suppressing the abundance of PLD1 in our bodies or in a patient's body, allowing them to undergo chemotherapy when needed.
All right. So to understand how PLD1 is directly linked with cancer development, MPO2 has split the process into three simple steps: hydrolysis, promotion, and overexpression. So the first step, hydrolysis, explains PLD1's ability to break down phosphatidylcholine into phosphatidic acid and choline. Phosphatidic acid then binds to the mTOR receptor, which starts a phosphorylation cascade. And this can be seen through diagram 1 on the next slide. All right, so different kinases use ATP to amplify the signal that mTOR has started. And once it reaches the centriole, mitosis begins. So PLD1 is actively low in mammals and is stimulated upon activation of the G-coupled protein receptors at the cell surface. As we can see through the diagram, GPCRs are involved in mitotic stimulations and stimulate mitosis by allowing cells to enter interphase. When PLD1 attaches as a ligand to the GPCRs, it repeatedly triggers a cell to go through interphase. Interphase consists of the G1 and S phase.
That's actually really sad. But I mean, like, maybe this is better for them.
The G1 phase.
Wait, did the blonde girls?
I'm sorry, could you go on mute? Okay, thank you. The G1 phase consists of the cell growth phase, and the S phase consists of DNA synthesis. So the cell that initially had the overexpression of PLD1 due to a carcinogenic mutation will continuously conduct in mitosis now. And this can be seen through diagram 2, which displays the results of an overabundance of PLD1 in the body. Kavya, the next slide. All right, so as I stated before, PLD1 is heavily known for its ability to use hydrolysis to break down phosphatidylcholine into phosphatidic acid and choline. Now, with so much phosphatic acid now being produced, it repeatedly triggers the mTOR pathway, prompting excessive and unhealthy cell mitosis. Excessive mitosis due to PLD1 links to potential cancer development. And so here's the final question. How do we prevent the overabundance of PLD1 to inhibit anti-apoptotic functions and tumorigenesis within cancer patients? The goal of this project is to create a competitive PLD1 inhibitor that can halt phosphatidic acid overproduction and stop cancer at its root.
That brings us to the solution we propose, and that is a synthetic aptamer. So before I explain the benefits of it, let me just talk a little bit about what an aptamer is. An aptamer is a strand of nucleic acids, usually a DNA or RNA molecule. And what they do is that it can act as a binding method. And in our case, we're going to be using it as a competitive inhibitor. So the reason why aptamers are a really good option is because they are safer and more budget-friendly avenue for treatment, meaning that there'd be significant cost reductions for patients. In addition, this is because they're cheap to design and manufacture. They're also less likely to be rejected by the immune system, which is very, very important for a treatment to work properly. In addition, they're also a larger range of target molecules, meaning that there's increased accuracy. And finally, they're easy to modify and they're more stable, all of which are very, very important for a successful solution. The importance of an aptamer and the importance of our project, well, it allows for more treatment options in the sense that it can allow patients to remain on chemotherapy rather than forcing them to switch to the more costly counterpart, which is immunotherapy, which, as is not already mentioned, is pretty inaccessible because of how expensive it is. In addition, there are issues with chemo and immunotherapy. For example, in prostate cancer, which is one of the cancers that our aptamer could potentially solve for, chemotherapy can have dangerous effects such as nerve damage and blood clots. In addition, there can be autoimmune reactions in immunotherapy because it basically causes the body to attack itself. And this can lead to things like neuropathy and more. As mentioned previously, there is CAR T cell therapy, which is an upcoming method, but it is still quite experimental and has a lot of adverse effects that haven't been properly explored. So an aptamer is a really good option at the moment, and that is why our project is quite centered on this. The expected outcomes would be that the aim of our project is to create a synthetic aptamer that will serve as a competitive inhibitor for the PLD1 enzyme. How this works is that it prevents PLD1, which is phosphatidylcholine. It prevents it from breaking down into phosphatidic acid in choline. As previously mentioned, what this does is that it prevents the mTOR and CRAF pathways from activating, and that in turn reduces excessive cell proliferation and tumorigenesis, because cell proliferation leads to tumorigenesis. If you look to the diagram on the right, you can see this kind of how this would work. So the 3D structure is formed and that creates the functional aptamer. This then uses target binding to bind to the biomarker or the active site of the enzyme. And it stops anti-apoptotic functions, which is essentially when the cell isn't able to self-destruct and leads to things like cell proliferation. The expected outcomes would be that we would have a synthetic aptamer that's able to regulate the expression of PLD1 while still being cost effective. So we kind of, solve for both major issues. And this would make it a lot more accessible to those who are affected, because accessibility is definitely one of the things that we would be solving for.
Okay, so now we're going to be talking about our methodology and the different steps we've taken in order to approach our goal. Next slide. Okay, so first we did electroporation. So we used electroporation to transform PLD1 into BLD2 electrocompetent cells. So essentially in electroporation, what's happening is that an electric shock is administered to the cells, and this temporarily increases the permeability of the cell membranes and makes it easier for foreign DNA to come into the cells. So essentially we use electroporation in order to get our foreign plasmid into our cells, making them BLD2 electrocompetent cells. Next, we did batch culture growth. So we put our single colony PLD1 sample, we put it into 3 different conditions of varying temperatures, and we grew it overnight with antibiotic ampicillin and TB media overnight. And then we also grew it in autoinduction media to catalyze the production of the electroporated sample from the previous step. The purpose of this is to later determine which of these temperatures will produce the greatest amount of PLD1 enzyme. Next, we did protein purification using nickel NTA columns, And this is a really essential step because it helps eliminate the possibility of contamination in our experiment. Okay, so next we did mini prep. So we used a mini prep kit to perform mini plasmid mini prep, and then we later did a toxicity test. So the mini prep is used to attain the plasmid, and the toxicity test is used to compare how different temperatures affect the density of our target protein, or like the amount of our target protein, which in this case is PLD1. Next, we did an SDS page gel. So we used tris acetate gel to suspend the protein in MES running buffer, and this protein is separated based on its kilodalton range, and this will indicate the presence of PLD1. So essentially what's happening is that the SDS page gel results in bands spread out throughout the gel, and we can compare it to our standard protein ladder using that as a ruler to determine which kilodalton range our protein falls under. We already know the kilodalton range that PLD1 falls under, and by comparing the known range of PLD1 with the one of our protein in the experiment, we can see if they match up. And if they do, this will indicate the presence of PLD1. So over the last few months, we've been working on isolating and purifying PLD1, which we've done successfully. But we have hit multiple roadblocks along the way, the biggest of which is contamination of our samples. You'll see this in the next few slides, have been able to verify the presence of PLD1 while also seeing contamination. And in order to combat this, we've been taking many precautions during our procedures, as well as following lab safety measures throughout the entire project.
Now on to our results.
Oh, they're more than halfway done. I think I missed Kavya's.
So onto our results. As you can see through the two STS gels pictured here, our gels contain the purified PLD1 sample in the first gel, and they are both within the 100 to 125 kilodalton range. However, wells 4 and 11 indicate potential presence of contamination within PLD1. Additionally, in our crude extract of PLD1, we also see that it's within the ideal 100 to 125 kilodalton range, but there is potential contamination. This leads us to our future work. To prevent further contamination, we are conducting plasma toxicity tests to determine at which temperature and with which media we can generate the highest protein output. Once we isolate the protein at the ideal point, we will move on to using selects to generate our synthetic aptamers. This will provide us with a series of potential candidates that we would then test using enzyme kinetics and protein modeling. Once we, what we're looking for in these steps is to see the upregulation of anti-apoptotic functions so that we see less cell proliferation and thereby less PLD1 signaling. In the end of all this, we would start by doing in silico structures of competitive inhibitors to create more products for later development.
And that brings us finally to our conclusion. So starting off with contamination, which was one of the bigger issues, in wells 4 and 11, there were bands that were shown, and we did see the presence of PLD1 protein samples within wells. However, it is unreliable for future analysis due to the possibility of contamination during procedures. And we think that this is most likely to have happened during mini prep and protein extraction steps, so in the future we will be more cautious when repeating these processes. Then when you look at SDS page gels, as seen in the gel, the bands with the 100 kilodalton to 150 kilodalton range appear less visible than the bands under the 75 kilodalton mark from the standard protein ladder, indicating potential either denaturation of our protein or there was just some sort of issue that in our future SDS page gels we'll be more cautious with. Additionally, the PLD1 crude extract sample had contaminations due to a lack of purification. However, our newly isolated crude extract samples indicate a presence of PLD1 protein. And finally, to kind of eradicate contamination, one of our biggest problems, we are going to be repeating mini prep and protein isolation, which is especially important because our next steps include testing gene toxicity by performing a plasma toxicity step.
We would like to thank ASDRP for allowing us to access their lab and resources that have made this scientific project possible. We would also like to extend our gratitude to our advisor, Mr. Amadi, whose guidance and expertise have been irreplaceable throughout this entire project. Thank you to all the participants and ASDRP staff whose insights, help, and time have made our project possible and expanded our growth as young researchers. Here are our references. And at this time, we'll take any questions and we'd be happy to answer them.